Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is October 31st, 2024, Halloween here in San Antonio. And we are talking with Raju Metherate, who is professor at the Department of Neurobiology and Behavior at the University of California, Irvine. Raju studies brain processing of sounds, especially how the auditory cortex contributes to the sound perception and processing, and the synaptic and neuron and circuit levels in the cortex to make that all happen. And he has a special interest in the cholinergic innervation of the cortex and how it affects the function of auditory pathways. Welcome, Raju.
Thank you. I'm happy to be here.
Also with us is Alfonso Apicella,
Hello.
a local auditory cortex expert.
Sorry, hello.
Hi. And me, I'm Charlie Wilson. So, in case somebody listening to us doesn't know all this, acetylcholine is an important neurotransmitter that's released in the cortex by axons of neurons that reside outside the cortex and are mostly not from the places we think of as the main pathway of the cortex. So in auditory cortex, they're not coming from the classical auditory pathway, but from other brain areas. And there are a lot of different acetylcholine receptors. They're in two major classes called nicotinic and muscarinic, and there's numerous ones of each of those. And they're distributed on the various cell types of the cortex, of which there are many. So it's quite a complicated thing to sort out. And people have liked to come up with a sort of simple idea about what acetylcholine does, like acetylcholine is about brain state and how awake you are, how attentive you are, something like that. But of course, the variety of receptors and the variety of cells makes it unlikely it'll be something that straightforward. It's never like that anyway. And then the nicotinic receptors are also the sites of action of nicotine, which is taken by a lot of people for whatever reasons they have for doing it, and it has profound effects on cortical function, and not all of which are bad. Mm-hmm. And although, nicotine we're mostly down on nicotine, but not necessarily for all the time. And Raju, could you start by telling us what nicotine does to our auditory cortex and on auditory perception?
Sure. So I have a lot of thoughts about this. Some of them are backed up by data and some aren't. But I would start with kind of what you just said about nicotine is being taken by people for specific reasons, and not all of the reasons are bad. So my crude way of thinking about acetylcholine and nicotine and brain function is that there are two main functions for acetylcholine in the brain, at least nicotinic acetylcholine receptors. One is in the cortex, and that mediates the effects of acetylcholine on higher cognitive functions, such as attention, and the other is in the reward pathways. So nicotine, the drug that mimics acetylcholine at nicotinic acetylcholine receptors, that's acting on both of those systems. When it acts on the reward pathway, that's why people are addicted, and that's a bad thing. And when it acts on the higher cognitive system, attentional system, that leads to positive effects of nicotine. And I should also add that I think, and other people think, that if you are addicted to nicotine per se, that's not necessarily a terrible thing. What's bad about being addicted to nicotine is that people get it in the form of tobacco. And there's all kinds of stuff in tobacco that will kill you. So that's a bad thing. But associated with that habit is probably enhancement of cognitive function, which is good.
So are there people who take nicotine in some other form other than tobacco for purposes of enhancing cognitive function, or are they just addicted?
Well, so that's a good question. The people who are trying to stop smoking will wear a patch or chew gum, a nicotine gum or a nicotine patch, and they will say they don't really like the gum or the patch. The only reason for using it is to stop smoking. So that plus experiment studies on non-smokers who are using gum or the patch for other reasons suggests that it's not addictive. The nicotine in gum and patch is not addictive. And it's probably not the nicotine itself, it's probably the method of delivery, the rate of absorption of nicotine.
I see. So nicotine's action on the reward pathway is still functioning somehow, but it doesn't seem rewarding.
It's not exactly clear. First of all, as a method to stop smoking, the patch and gum aren't terribly effective. It doesn't work for a lot of people. And possibly it's whether or not the nicotine is addictive or not has to do with the rate and dose that reaches, the rate at which it reaches the brain, and the dose, the concentration that reaches the brain. And it might be because of the types of nicotine receptor subtypes that are in the reward system versus others.
And it's known that when they stop, the effect as a decline in cognitive function, or is just basically when you stop, correct, nicotine via tobacco, there is other mechanism they get involved, and this is why, it's a controversial between the decline or the fact that you are not smoking anymore. Is there any evidence for that?
So the cognitive enhancing effects of nicotine were first demonstrated on smokers who had stopped smoking or who had who might be continually smoking but didn't smoke prior to the test. And so it seems clear that the enhancement of cognitive function in those people seems to be a relief from withdrawal. So yes, that is an issue. But more recent studies have shown similar effects or cognitive enhancing effects in non-smokers. So that doesn't have anything to do with withdrawal.
So it's pretty vague, enhancing cognitive effects. In fact, it's kind of subjective. I might think my cognition is improved when it's not. But what evidence do we have in the auditory system for enhanced function with nicotine?
Yeah, so I would have to remember those studies. The studies are pretty clear. I mean, this is an interesting phenomenon, that the studies seem pretty clear in animals. There's a whole variety of tests there where animals perform better when they have nicotine on board. There are similar findings in humans. The results are much more subtle. And you look at the results as someone who is used to looking at animal studies, and you think those are pretty weak results. They're statistically significant, but they're not that impressive. But there's a whole bunch of statistically significant findings.
So those kinds of enhancements are like, I can hear sounds that would normally be too quiet for me to hear, or I can distinguish the difference between two frequencies I normally would have trouble distinguishing between them.
It's most, I think, like distinguishing different frequencies. A lot of the studies are visual. There are fewer in the auditory system. Auditory system, I'm thinking like novelty detection. That people will be the kinds of ERPs that are generated by novel stimuli, larger amplitude, lower threshold, things like that.
I see. So, is there a potential? It sounds so subtle. I would wonder if there could be a actual clinical use for that in improving anybody's cognition.
Right.
But you've thought about it being helpful with people who have auditory problems. What kind of problems would those be?
My understanding as a non-physician is that central auditory processing disorders applies to a whole bunch of things. Basically saying that your hearing problem is not in the ear, it's in the brain. That's the main thing. Within that group, there seems to be a whole different. I've looked at the tests that a psychologist would use to try to tell what kind of auditory processing disorder you have, and they encompass a whole variety of things. can you hear? First, can you hear? An audiologist does it very simple. These people can all hear. That's fine. Their thresholds are fine. Can you understand words? That often is fine. The kind of thing that I'm thinking that it's applicable to is, can you not just can you understand sentences, but a whole bunch of instructions and sentences sequentially? Can you process them all in an effective manner? Can you build on one sentence and get the next one and build up an understanding of what is being said? Kind of like what you would get in an experience in a classroom or a seminar, something like that, where a logical argument is being laid out. That is a pretty subtle effect, right?
Yeah.
It's hard to have a test that will capture that.
But everybody knows the feeling of not doing it well, right?
Yes.
I know
yes.
this is all making sense. It's coming at me pretty fast, and
Yes.
I'm losing the thread.
Right. I understand every sentence you're saying. I'm not building the argument in my head.
So you're saying when that happens to me, I might consider chewing a piece of nicotine gum?
Possibly.
I'll take that under consideration. So tell us what it's doing in the auditory cortex at that time. And there are other places, there's acetylcholine in other places in the auditory pathway too, right? So it's probably working at several different levels.
Right.
But do we know more about the cortex than anywhere else? It's been studied the most.
Yes, I think so. Due to a few labs, people have looked at using EEG caps where they can see auditory brainstem responses as well as midbrain responses and cortical responses. And people are seeing that when you put a patch on non-smokers who are being studied this way, the patch seems to affect the long duration, long latency potentials, whereas it's not affecting the auditory brainstem responses. So the input from the ear and the brainstem responses are not being affected, whereas the longer latency the potentials are cortical potentials, but not brainstem potentials. This is a little hard to understand because there are nicotinic acetylcholine receptors everywhere, from starting in the ear. The brainstem is full of them, the midbrain, everything. There's receptors everywhere. It's not exactly clear why this particular experiment is showing an effect on the cortical responses and not the earlier responses.
Yeah, acetylcholine pathways are like dopamine and norepinephrine. They just go everywhere. There's almost no place that doesn't have some acetylcholine innervation and acetylcholine receptors.
Right.
I think that makes it kind of hard to study in detail, but it also is what makes us think of it as brain state signal rather than fine detail about the world kind of signal.
Right.
And that's probably jumping to conclusions.
Do you also think that the difficulty is because the different distribution of different subunits of the nicotine receptor along the auditory pathway?
Yes.
And across different cell types too, correct?
That surely matters. Like the different subtypes of nicotinic acetylcholine receptor require different concentrations of nicotine or acetylcholine to activate. So it's possible, and they desensitize to different degrees. Some of them desensitize within milliseconds, others take seconds or minutes to desensitize, and some possibly don't desensitize at all. So when you give a systemic administration like a patch, that may not be enough to activate, say, the nicotine, the alpha-9, alpha-10 receptors in the ear, but it could activate the alpha-4, beta-2 or alpha-2 receptors in the cortex. So
sounds like an opportunity for pharmacologists who are looking for specific agonists that would do one thing and not the other.
Yes.
So in that case, the complexity of the cholinergic system becomes an opportunity from a drug design sort of point of view.
Right. Certainly the drug companies feel that. They're always trying to build selective agonists for use as cognitive enhancers or treatments for dementia, things like that.
But any of those work? Any of those make it to market and are used?
There were, I think 10 years ago, last time I looked, there were a number of clinical trials. As far as I know, most or all have failed. I'm not entirely sure why or what happened. I remember talking to one of the scientists at Abbott or AbbVie, who thought that the drug that he had developed was first of all, they got to clinical trials because they were very effective in animals. And he thinks there are good reasons why it still works, but it failed a clinical trial. That's outside of my expertise. But
so maybe, I mean, if we knew which receptors was on which neurons and which neurons are responsible for which effects, we could kind of reason it out, what you would want. So
yes.
could you give us a little rundown on what we know about what happens in auditory cortex neurons, their responses, and how nicotine changes them?
Well, the finding from our lab that I'm most excited about is the newest stuff I was talking about earlier, which is that alpha-2 receptors may be important for the cognitive functions. Alpha-2 receptors are also found in the reward pathway, so I'm not sure it's just cognitive and not reward. But in terms of cognitive function, the alpha-2 receptors are relatively rare, very rare, maybe less than 3% of the nicotinic acetylcholine receptors in cortex, and they seem to be found on one specific cell type, and they do not desensitize. So when you give a systemic administration of nicotine, it's possible that all the other nicotinic receptors that are doing these other things on other cells, they might desensitize, leaving this one cell type continuing to function, and maybe its function would dominate over the functions of the receptors that have desensitized.
And do you think that the same thing will apply to the reward system that express the alpha?
It's quite possible. We haven't studied there.
Then it still becomes a dilemma to dissect what is the cognitive versus the reward component, correct, of these enhancements.
Right.
Anything that you can tell more about really how the nicotine affect the underlying circuits in the cortex and the behavior of the animal, correct? There is a lot of study about acetylcholine that have an enhancement in plasticity and therefore learning. What do you think about the effect of nicotine in the auditory cortex? And this cognitive enhancement is also due to a better learning or discrimination behavior? What do you think?
Yes, so what we see in auditory cortex is that the auditory cortical cells are all tuned to sound, and the cells have a frequency tuning curve, so they're responsive to some frequencies and not others. And we see that when we give the animal nicotine systemically, that tuning becomes sharper. They're more attuned to a narrower range of frequencies. The circuit in auditory cortex that we think is mediating nicotine's effect on these tuning curves, that circuit is seen throughout cortex. But of course, outside of auditory cortex, it's doing something else. That same circuit is doing something else. Just the way that the auditory cortex is taking inputs about different frequencies and forming them into a tuning curve, the same circuit in other cortical areas is taking information about something else. And when we give nicotine, we think that tuning will be enhanced for that something else, whatever it is that that cortical region is doing. So we think the auditory cortex is a model system for what nicotine is doing throughout cortex. We just don't know what all those other functions
so
are.
For example, our ability to tell two frequencies apart has to do with how broad those tuning curves are. If they're too wide, we can't tell them apart, just like two stimuli on our skin that are too close together.
Right.
So we do that with all of our cortex, thinking about two thoughts and how related they are, and probably in a way.
Yes.
And
But the question that I have for you, correct? Yes, is changing, the way how the cell respond to the single frequency. But your idea also, as you start, that the problem is not only per se to know what is a single frequency is, but to put the piece of the sentence together, correct? How the cortex then does that, correct? When we have, correct, each single cells right now respond to a single frequency, and they're trying to put all together, because also the auditory cortex, I don't know if I can say, but it's a mess, correct? We know that each layer, correct, they don't respond to the same sound, even when we do a penetration that goes along the same, axis. What do you think, correct? How right now this enhancement of the single frequency will explain how we put piece together of a sentence to make a coherence of what the thought is?
Or even to find a word. Because a word has got a bunch of different
correct.
sounds in it.
Yeah. Well, so these are great questions, and we have no idea. The problem is that we're thinking about this problem from two perspectives. One is the bottom up. How do individual cells in a dish respond to an excitatory input? How is that response to an excitatory input modified by a drug like nicotine? And on the other side, we're looking at humans doing some task, listening to a sentence and asking, well, if we put a patch on them, what changes? And we're, coming at this from a bottom-up approach and a top-down approach, and we have no idea how they're going to build. At some point, we believe, we're scientists, we believe we should be able to explain that top-down phenomenon in terms of the bottom-up explanation, but we're not there yet.
I think part of it boils down to, what do you think the primary auditory cortex does? Because probably the primary auditory cortex is not the place that puts a bunch of words together into a sentence or puts a lecture into a thread of thought. It's probably doing something simpler with sound.
Right.
And but how far does it go? Does the auditory cortex analyze words, detect words, or does it even like this? I know the auditory cortex ought to be able to detect notes, but could it detect a chord? And it seems like a cortical thing to do.
There are evidence that show that the cortical neuron are able to detect chords, correct, has been used in a lot. So this is, that was my next question, correct? I was not thinking about chords, correct? But I was thinking, once again, rather than see only how the characteristic frequency change, can we see, correct, if there are sequence of sound, and see then how the nicotine affect, correct? Even in an oddball situation where we can use frequency modulation up, and then we have an oddball that is frequency modulation down, correct? Or how nicotine has an effect on history-dependent phenomena in the cortex, correct? And anybody has done any study like that?
Not in terms of nicotine, not that I can remember, but there are people who record from auditory cortex and human cortex patients who are listening to sentences, right? So the auditory cortex does respond to individual words and sentences. It's not really clear to what degree it's contributing to comprehension, but people do see changes in the auditory cortex response to words and sentences with levels of attention. For example, it tracks the contours of the words and sentences better when you're paying attention to it. That resembles the effects of nicotine that we see on sequences of sounds, of pure tones, for example. And it makes a lot of sense given what we see about the effects of nicotine on cellular synaptic activity, that nicotine should help cortex track sequences of sounds better.
So the nicotine receptor that you think is the most important because it doesn't desensitize is on an interneuron whose job it is to inhibit a bunch of other interneurons to let the pyramidal neuron fire, something like that, right?
Right.
So can we think about that in a sequence, like we normally do experiments where we say, I'm going to make that interneuron fire, and then I'm going to see what it does to the pyramidal cell right now over the next 25 milliseconds or something like that. But if we imagined a sequence of stimuli, can we see how that, Alfonso, this is what you do, right?
Yeah.
Can you tell us how an interneuron like that, a disinhibitory interneuron, could play part of the recognition of some kind of a sequence, or alter, given a different response to a sequence than to the individual piece?
The problem that, when we get in the world of interneuron, it's a very complicated, correct, when we look at the sequence, correct? Because, we know there is at least 3 different classes of GABAergic neurons in the cortex, and we know that they differentially respond to sequence. There are, for example, neurons that are trying to adapt their response, correct? They get less responsive during the sequence of the presentation. There are some that enhance their response. And there are some that do both of them. And also, these interneurons, they differentially, not only target different compartments of the pyramidal cells, but also they differentially innervate each other for this sort of disinhibitory mechanism. I have right now not any study on the top of my mind that have, a clear explanation how different GABAergic neurons really work on the sequences of sound, correct, and make sense of a word. I don't know if Raju, you have any study like that says anything that how is the role of interneurons on the sequence of words?
I think we're held back by the history of how we did experiments. We give an electrical shock here, and then we see what happens there, and then maybe we'll give a train of electrical shocks, and that's the closest thing we have to
I think the thing that I would like to do, and this is, it's rather than give and train. I know it's something that I'm putting on hold for a long time. We have discussed about that, correct? I would like to use sinus wave, because after all, tones are sinus wave. I would like to see how different sinus wave will lead to different response of the GABAergic neurons, correct? And maybe, not even that. I will, a sequence, this is what I thought, and this is, I don't know if it's a great idea or not, but a sequence of tones are made of different frequency, correct? Is that possible that we can make some sort of what we call in electrophysiology chirp, right? A sinus wave that change, in the frequency over time, and see how these different class of GABAergic neurons respond to that, and probably maybe right now to see how nicotine enhance or change their response.
So it's really common to give a frequency modulated sweep.
Yes.
Which is like a chirp, kind of. It goes one direction, starts at some frequency and ends up at another one. And people give those all the time. Do neurons respond differently to a rising one versus a falling one?
Yes. I just, I don't know. You read my mind, because these are the papers that I have on my desk right now. And yes, we know there are, just to look at the pyramidal neurons, we know there are neurons that prefer upward sweeps, are called, correct, and downward sweeps. And particularly, I think there is an old study from Li Zhang that he saw that neurons that have a low characteristic frequency, they prefer upward sweeps compared to neurons that have, high characteristic frequency to respond, to downward sweeps. When you look at that, correct, the thing, it starts to get complicated, correct? It's like visual discrimination, correct? Or, is what is happening, correct? We have 2 frequency, and either they are going to have a summation effect, correct? During the sweeps, you have multiple frequencies. Therefore, either you have one input that is coming first, the other that is coming second, but they can both, during the sweeps, they can add to each other to generate a stronger response.
I see. So we're thinking about the different frequencies as being represented by different inputs to our cortical neuron.
Correct.
Right. So as we do a sweep in frequency, we're stimulating different thalamocortical cells
along their favorite, direction of the sweeps.
So it's not an intrinsic property of the cell that it likes that sweep.
Correct.
It has to do with the wiring of that cell to some prior station in the auditory system.
Yes. But then when you go in the null direction, correct, when the nuance is not this preferential direction of the frequency modulation, you see basically that the timing of the two input will lead, to 2-week response. This is what I'm telling you, and this is what we'd like to do in the future. This is without counting that there is also inhibition, the so-called sideband of the inhibition in the auditory field. Because then what is going to happen that when you have the two tones in the sweeps in the right direction, correct, you have still an excitatory effect. But when you go in the opposite direction and that frequency lead to inhibition, correct, the sideband inhibition, basically you will have, a decreased response. I'm telling you, the synaptic mechanism of this frequency modulation are very interesting. On the top of my head, and please, I'm sorry if I don't know anybody yet that is really look at what is the role of the 3 classes of GABAergic neurons on this effect on the sweeps, on the frequency modulation.
So it could be incredibly difficult to figure out, because it could be that they all get different groups of inputs from different thalamic neurons, but it's likely that they're near each other and they're getting similar synaptic inputs, and that the local circuit is what's determining the specialization of the neuron's activity, or?
I think it can be both. I don't think that is only because, one.
Because I was just trying to figure out what would happen if we just took one of these interneurons and put alpha-2 nicotinic receptor on it and gave it nicotine. Would we be able to predict an effect? Because that's, we would have an experiment.
I think you did one experiment like that, correct, where you removed the alpha-2 from one of these interneurons, correct?
So we removed alpha-2 from the brain.
From the brain, sorry.
It was a knockout mouse.
Okay.
But in cortex, it seems to be on one kind of cell, the Martinotti cell.
Okay, therefore we know the effect.
So what was it?
It's the Martinotti neurons in the layer 5.
Yeah, but then what?
So the effect that we've been studying is how nicotine makes receptive fields narrower, and in the knockout mouse, it did not. So we think the Martinotti cells are regulating the width of the tuning curve.
I see. Okay, so it's not just strictly connectivity; it's the dynamics within the circuit.
Yes, right.
How they do that? What is the mechanism, correct? Because
Martinotti cells do that?
Yeah. We know that the Martinotti cells, correct, preferentially have these dendrites, correct, in the layer 5, and then they have the axon that goes all up there, and they go most of the time, correct, it's on the distal apical dendrites of the pyramidal cells, correct? How they do this kind of announcement? Because at this point, it needs to do something with synaptic integration of inputs along the dendrites, correct?
What would it be the effects on the interneurons that it's contacting on the way up there?
This is the question,
so
correct?
There are at least 2 types of Martinotti cell, one that goes only to layer 1 and one that goes to several superficial layers. And only a subset of the ascending axons from Martinotti cells contact interneurons. Some of them are directly contacting the apical dendrites of pyramidal cells. So if you're looking at the pyramidal cell, the effect of the Martinotti cell activation is to inhibit the distal apical dendrite and then disinhibit the more proximal apical dendrite. And we think that the inputs that are controlling the width of the receptive field are to those distal apical dendrites. So they're being inhibited preferentially. So that's why the receptive field gets narrower when you activate the Martinotti cell. That's the hypothesis.
And then, once again, this is my curiosity, how they do that, correct? Because when there was a beautiful calcium imaging experiment, correct, that when you look at the dendrites of pyramidal cells in the auditory cortex, basically the spine can respond to all the frequencies. Now, would you think that this, the narrower of this receptive field, how the Martinotti cells making an inhibition on this part of the dendrite is going to lead once again to the enhancement? It's a mechanism that you think it's, like you mentioned at some point, a filter basically, the input, all the frequency arriving on this spine, but basically the Martinotti cells are going to inhibit branches. They don't need to listen to that particular frequency and enhancing all that, only that one. I don't know, correct. I'm thinking about, correct. Somebody needs to do the job because, the beauty is that when you look at all these calcium imaging experiments, correct, in the auditory cortex,
Right.
sometimes we dispute what is the tonotopic organization because there is all this salt and pepper there.
Right.
But the beauty was there is this experiment that I found interesting because basically all the pyramidal cells, they have all the neurons, correct, in the auditory cortex, have the possibility to listen to all the frequency they want. But then how they decide, correct, to get tuned to a particular characteristic frequency, and on the top of that, the nicotine, it's lead to a gain, correct?
Yeah.
What do you think are the mechanisms that is happening?
So you're talking about very controversial, not controversial, but exciting, but not fully understood finding how multiples, if you look at spines on pyramidal cells, they have they respond to multiple frequencies. So I don't think it's known what inputs are generating those multiple frequency responses. It could be that they are a combination of inputs directly coming from the thalamus, thalamocortical synapses, as well as intracortical horizontal inputs. So what we're thinking of the Martinotti cells doing is selectively inhibiting the horizontal inputs. So the same spines might be getting multiple inputs, but one of those inputs the horizontal inputs has been shut down somehow.
So it seems to me all we have to do is figure out the cortical circuitry and how it works. And then we could design a nicotinic agonist to make us super processors of auditory information.
Make it high cognitive, how the
I think you're right. That's all we have to do.
This means that I will be able to hear 60 kilohertz frequency or
follow an argument better.
So thanks a lot, Raju. It was really great having you here today.
Thank you. It's fun being here.
Thanks, Alfonso.
Thank you, Charlie. Pleasure.
It's been Neuroscientists Talk Shop.